Once again, I want to welcome every one of you. This is going to be the third day on our training of a complete beginner's kit, starting from zero to the professional level. That is moving from wet lab to the dry lab. We actually started some days ago, and we were able to look at the workflow. Once again, the workflow, we ran through it, pre-sampling and some other things, and we actually got to the galaxy level. And at that galaxy, I think the last thing we did was using spade. Abi, using spade. What do we use spade for? What did we use spade to do? For genome assembly. So that is, you assemble them. We started with looking at, I said the first thing you should do is to do the data cleaning. And that is finding the quality control. The quality of your data matter most. And that's the reason why the first thing you do is to make use of a FastQC. And at that point, we were able to see that our data was ready to be used for further molecular processes or bioinformatics processes. Don't forget that I said whether we are going to whichever subgroup or cohort group that you belong to, whether you are in drug design group, antimicrobial resistant group, or vaccine design group, what is most important is to be able to have a complete sequence where you are going to have adequate information about your organism. And don't forget that in this group, I said you should upload and make sure you upload data that is on TB, that is Mycobacterium tuberculosis data, and which I think you have already uploaded, and which I believe you must have done that. After checking the quality of your data, and I told you one of the reasons why you must check the quality is to be sure that your data is free from contamination or contaminants. Then after that, you now use Trimomatic or fastp or cut adapt for what? For trimming. You need to trim your data, especially the adapter. So you need to trim them. Now, I want you to look at the trimming, or what is called adapter. I want you to look at it as if you are looking at a magnet. You will see that when you have a magnet, and the magnet is what you want to use to fix out a needle in a heap of sand. You are looking for that needle, so we call it probing. You are probing that sand of magnetized ion that could be attracted towards your magnet. Not only that, you will now discover that apart from your needle, there are some other ion filings, small particles of ion filings that can be adapted to your magnet. So the idea of trimming using Trimomatic is like you are removing all those ion filing that have been adapted or attracted towards your magnet. So that is exactly what trimming procedure is all about. Now, after you must have trimmed, I now made mention of you have two options of running comparative genomic analysis about your sample. Number one, it is that you already have a reference species in which you know everything about that species. And I told you that in the case of Mycobacterium tuberculosis, the um, reference species is called H37RV. So you already have that. That is what is called reference comparative genomics. And you can also do what is called de novo assembly, whereby you only focus on your own data, your own sequences only, and you won't compare it with any other any other sequence. So that is very possible. And I said, if you want to use reference genomics, or what we can call reference mapping, it's also called reference marking, mapping, is that you are trying to see how very, very important, or how very similar is your sequence, or how very unique is your sequence to the overall, which is the reference. So, and what you will do is that I gave you a tool that you use BWA MEM2. You use that for alignment. And don't forget that that day I also cut out or cut out part of the interpretation of this area, and I pasted it on the ChatGPT that actually told us and made some emphasis about the interpretation of your result. And just like I told you that once you are confused, anytime you get confused, wherever that point you are, you can just screenshot it and tell ChatGPT that this is the point you are. What is the next thing it is going to put you through? So, and that's why that as a student of bioinformatics, as a computational biologist, you need to get acquainted, not even familiar. Acquaintance is different from familiar. Familiar is I know your face now, we might not be friends. But when we are talking of she is my acquaintance, that is, you know everything about that person. So you should make ChatGPT to be your acquaintance. That whatever, in fact, sometimes you will get used to ChatGPT that even if you do not prompt it, it's already aware of you. Huh? Even if you do not type your name, you do not type anything, just go straight to the prompt you wanted ChatGPT to run for you. It's going to go back to the memory and go and retrieve every data about you, and it's going to use that to prompt. So that is the reason why ChatGPT, honestly speaking, me, in fact, it has become like pure water. Sometimes you might want to even eat in your house and just say, see, I don't want to develop ulcer. What are those foods that I can eat? Can you recommend to me? ChatGPT will tell you. So he's a very close, in fact, the closest companion. So anytime you get confused, anytime you seem to be alone, and that's the reason why I said that number one, you listen to the YouTube, then sometimes you can get confused, sometimes you can get lost. So just ask ChatGPT to just follow. So if we say that you should use this tool, and as I told you that bioinformatics, you need to be versatile. There are some tools that may be available by 4 o'clock, and by 6 o'clock it may no longer be available. So that's the reason why. So when you now run at that um, circumstance, the best thing is you need a guiding angel. You need somebody that can actually put you through. It's just like your comforter. Oh, I am so tired. I am confused today. I don't even know what to do. My supervisor is all over my neck, and I don't know where to go to. I need consolation. It's going to console you. It's going to console you. So the same thing. So that, as I said, you need to get acquainted. Sometimes you may see some things that you may just come across new tools that somebody just recommends for you that this tool can also be used to perform this task. Even if you have not used it before, you start playing around it. Read more about it, that ChatGPT should please do it for you, put you through how to use it step by step. Of And don't forget that sometimes ChatGPT is different from one of the differences between chatbots, that is ChatGPT, and human. Human can get bored. That your question is just too much. You asked me this yesterday, you are still asking me the same thing, and you can just get the rods. Please go away as if you don't know anything. ChatGPT will not forsake you. So sometimes if it give you an answer or response, if you are still not satisfied, you can still tell that I am not still satisfied. I want a beginner level response. It's still going to refine it for you. And I also told you that don't mind the language. Sometimes you might not have the command of English. You might not have, sometimes you will have something that you want to do something in your head, but in your mind, you don't know how to go about it. So that one, just pour everything. You can even pour it in Yoruba or any other language and ask ChatGPT to translate to correct English for you. It's going to do it. One of the easiest way, gone were those days that you labor for a long time. Now you can get a lot of things as much as you are available on internet. So just like I said, ChatGPT, we prompted ChatGPT that day, and he told us that alignment, from that word alignment, that is, we are trying to compare. You are bringing two sequences, your own, and you are trying to align it with others. That is what is called alignment. You want to look at the differences, you want to look at their similarities, and you want to look at the mutational changes that might have occurred in them. And don't forget that we are going to see it. We are going to see it. I have been telling you even in the theory class that, you see, in some time past, there are some places that some microorganisms are only restricted to that places. That anywhere you want to find them, you go to their real natural habitat. But nowadays, we now have so many outside their ecological niches, outside their function. So, and how are they going? I mean, how have they been coping with these new strategies? Or, which I said, it has to be a kind of mutational changes. They have to change. They have to adapt. So, microorganism is such that wherever they find themselves, and just like human being, wherever you find themselves, it may only take some time. You get used to it. But what microorganisms do anytime they find themselves in a new environment is that they will just very, very simple. They change their what? Molecular or genetic information. So why I'm saying this is just that mutation occurs, and every cellular activity, that takes place, be it physiological activities, nutritional, whichever one, they are very, very subject to changes. We call it spontaneous mutation. Spontaneous mutation is that it's a de novo mutation. It's a mutation that nobody triggered it. It's just natural. For example, now, I remember the way we normally understand in those days mutation in plants. That was the use of onion bulbs. So just put ordinary water. Just put ordinary water and you put the onion bulb on it, let the onion grow. By the time the onion grows, in normal water, put the same onion in the same solution, just discover that after specific time or minutes, you see changes naturally. And that's why some people still believe that up to today, human being, even our waking up every day and every time, in fact, there was an article I read that any bite by mosquito, any bite by mosquito on you, that mosquito has changed, will always inflict molecular changes on you. So that means you are no longer the same person before the bite of mosquito and after the bite of mosquito. So not to even talk about. So what we are saying in essence is that you needed mutation is something that you need to be cognizant of. You need to know that certain, according to Charles Darwin, which stated that survival of the fittest, that every organism is subject to what? Mutational changes. So that is one of the reasons why we normally use CB reference or any reference data for our, and that's what we call it mapping. You map it. So you use that tool. And we also mentioned some tools for BAM, sorting, indexing, and all the rest. So without wasting our time, because what we want to do, this one, I am not going to let you do what is called reference genomic analysis. What we are going to make use of de novo. That is, we are not going to compare it with the standard. So that's the reason why that let me once again display the okay. So reference mapping, I told us that we are not interested in that. So we'll now go to the next one, and that is going to be the number 7 in the flow chart. In the number 7, let me read once again. The number one is a project setup and metadata. The second one is raw read and a QC. That is where you use a faster QC, followed by read and trimming, followed by read and trimming and cleaning. Okay. After that, that's number 3. After that, we now look at reference mapping. Reference mapping of Mycobacterium tuberculosis, which is H37RV in case of that. Then the next one is a coverage assessment and mapping of QC. We are not going to need this because we are not going to make use of genomic reference genomic assembly. Then number 6 is variant calling and filtering, whereby you just come across the single nucleotide proteins, polypeptide. How can you identify SNP? So that is beyond here. So we now move straight to the next one, which has to do with TB specific drug resistance. TB specific drug resistance lineage prediction. So, and what is the goal of this? The goal is to validate the multi-drug resistance status. Now, let me tell you that the sequences which you have inputted in your data, that is the sequence of drug resistance. Drug resistance. So we now want to see that Mycobacterium tuberculosis, we want to see the whole genome, and we want to see what are those genes that Mycobacterium tuberculosis is using for multi-drug resistance. So this should be our interest. Our interest in the sense that I told us at the beginning of this workshop that you must know that any drugs, any gene that any micro organism is expressing, it is an essential gene. It is an essential protein. It is what you need. According to Jean Lamarck, postulated of evolution. Jean Lamarck postulated about the theory of use and disuse. What Jean Lamarck said was the organ or the attributes in which you are using continuously or constantly, you will tend to develop it more than the one do not actually need. For example, let me just use what the evolution, human evolution as an example. The human evolution, we still believe that, and we know very well that we are also like monkey. Our ancestor was an ape. That is, if that is true, that means the same way an ape or a monkey has tail is the same way we also have our own tail. And if you touch it, you will see that every one of us will have our own tail. But what do we need the tail for? We don't need tail. And that is why that organ that we do not use is now becoming degenerated. We call it rudimentary. But the monkeys, they need the tail. Why do they need the tail? They use that tail to be able to hang. They'll be able to use that to hang on the tree, jumping from one tree to another. Human beings do not need that. So we have to drop the one we don't need and develop on the one that we actually need. So the same thing with microorganisms. So any protein, so to say, any protein, they will tend to develop it once it is an essential protein for them. So, for example, the protein that is responsible for virulence, causing the protein they use in causing diseases, they will tend to develop it than the one they will use for another purpose. So that is one thing that you need to know. So in this TB, Mycobacterium tuberculosis, the first thing is we want to identify multi-drug resistance, and that is should be the interest of those that are going to design vaccine. If you are going to design vaccine, you are designing vaccine for multi-drug resistance. Gene, you will identify it. And those ones that are not drug resistant, you also identify it. And even this same set of protein that are essential protein, that are very, very important, that to the level that Mycobacterium tuberculosis use it to escape the host immune system. Now, let me tell you something. In immunology, under normal circumstance, Mycobacterium tuberculosis, for any immunocompetent individual, that is somebody whose immune system is very, very competent, we call them immunocompetent. That is somebody that can take care of any infection, that the infection will not be able, or the microorganism will not be able to establish itself in that host. We call them immunocompetent hosts. Under normal circumstance, one of the reasons why you are still like this, I told you that research told us that 90% of human population is infected with latent Mycobacterium tuberculosis. All of us, 90% of us, if we conduct that test on us, we will see, even if you use that rapid test kit, you will see that who will just be TB positive. But one of the reasons why we do not do that is just that immediately we inhale it, we have what is called the macrophages, which I told you in immunology that macrophages, they are professional phagocytes. That is, they swallow. So immediately TB comes in, it will go into the lungs straight away. When it gets into the lungs, it's going to encounter the macrophages. So the macrophages, what will they do? They will just go and surround the TB bacilli. They just go and surround it. So it's just like an armed robber that has broke into your house, and immediately the security now surround it and say, Mr. Man, you cannot go further. So the same thing macrophages will do. So by the time the macrophages now surround the TB, that is what is called the latent TB. Latent because it is resting. It is resting. It is there, but it will not manifest. It will not multiply further. Anyway, what you can just do is that, please, we don't say you should not enter our house, but don't move further. Any attempt to move further, we are going to deal with you. That is exactly what the macrophages will do. Now, do you know that sometimes I've told you that the macrophages have now they've surrounded the TB. So sometimes, do you know what that Mycobacterium will do? The bacteria will now go and do what is called variants. They will go and change, mimic. They will go and change their own codes so that they will now appear to be part of your cell. So that macrophages will no longer be able, for example, now when you surround, when your security now surround an armed robber, and now said the armed robber now said, see, why are you surrounding me? I am also part of you now. So, okay, what is the means of identification? And once he's able to produce that means of identification, say, yes, you can go. So the same thing with Mycobacterium tuberculosis. They have some proteins that immediately they are now being recognized by my professional phagocytes. They will now use that protein to be able to deceive the immune system, and the immune system will just allow them to go. Whereas they are the enemy. So once they now allow them to go now, they will now go and multiply. And that is the level we now call it pulmonary TB. If it retains in the lungs, pulmonary. Anything that has to do with pulmonary is talking about the lungs. And sometimes that TB will no longer stop. You see, when you allow an armed robbers to do anyhow in your house, the armed robber now said, please, just allow me to get into your sitting room. I don't have any interest in going to any other place. So you now allow the armed robbers. So the next thing is that, let me go to your kitchen. Let me go to your bedroom. Let me go to your deal. So until they have now overpowered you, they will still want you to take them to another house. Lead us to where there is money. So the same thing, by the time, and that is what we now call systemic TB. Systemic because it is now dealing with the rest part of the body. You can have a TB of the brain, you have TB in the kidney, you can find them anywhere because at that point in time, no more control. Your immune system is no longer in charge. And that's the reason why that everything that you need to be very, 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 very sure of is that there should not be anything that will weaken your immune system. By the time your immune system is weakened, the ugly TB will now rise up. That yes, we have been able to see the finality of our enemy, and that is we are now in charge. That is that level of that. So there are different mechanisms of doing that. I have told you that sometimes, when you see an armed robber also dressed like the hunter, also dressed like the security with the same gun, like a soldier that is protecting your immune system, that is protecting your body, the same antigen, the same enemy now dressed like that one. So how will you now be able to differentiate there. So you see, the story of that's why I said, even if you want to do the wet lab, you also needed to understand what, I mean, you want to do the dry lab, you needed to understand the basic mechanism, the basic biology behind the organism you are trying to either develop drugs, either develop drug resistance, or either develop what? Vaccine. Yes. So that's you need to know. And just like I said, we will now make use of one other tool which I have told you, TB profiler. I think I said that, Abi.
Yes.
TB profiler. So have you registered for TB profiler? You didn't register. So what we will do is that still in that galaxy, we have a TB profiler inside there. So that's the one we are going to make use for today. But the other one, the results that you have from TB profiler in the galaxy, you can run that one, which is a standalone TB profiler, and compare the results we are going to have, whether it is going to be the same or it's not going to be the same. Okay, so please let us now, what do we mean? What is the rationale behind the use of TB profiling? So this is a tool. This tool perform two roles. Two roles, or they perform two roles. Number one is that they will validate the multi-drug resistance. Yeah, multi-drug resistant, they will validate it. They will validate the gene, whether the gene is responsible for multi-drug resistance or not. Then apart from that, after validation, is now going to link it with the lineage. Is going to link it with the ancestor. Do you know that sometimes most of the TB we have here, their lineage could be in China. We are going to see it. Their own lineage could be in China, and they are found in Nigeria here. How did they get to this place? How did they get to this place? And that's the reason why we need a powerful tool that's going to tell us the evolution. Now, let me tell you one thing. The enemy, there are two types of enemy. One enemy is more notorious than another one. Which one is going to be more notorious than another one? The enemy that you know everything about them. You know their origin, you know the father, you know everything about them, and the ones you do not know anything about. Which one is going to be?
The one you know.
You know everything. It's going to be so easier to deal with. So that's the reason why if you want to develop drug, vaccine, or you want to put up resistance, surveillance of any, once you are able to know the evolutionary lineage or the evolutionary tree or evolutionary history about that, honestly speaking, you have succeeded to certain level. So that is what TB profiler is going to do. Two, I said the first one is that it's going to tell you some information, whether it is a multi-drug resistance or not. Then the second one is going to tell you, classify your organism or your sequence based on the evolutionary lineage. So that is what a TB profiler will do. And the tools that you can use to do this, TB profiler, and you have the website, the link over there. And the second one is a microbe predictor. Microbe predictor. And what will you do? If you are using this, you just run TB profiler on your FastQ, TB profiler on your FastQ. Don't forget that at this level, the where you started with, the raw data that I asked you to to install, is what you will use for TB profiler. not the one that has been trimmed. Do you understand? Not that one. So you need to actually know that. And the output, resistance conferring mutations. These are examples of mutation you can see in Mycobacterium tuberculosis. RPOB, CAT-G, these are gyrase and so on. All these are the name of certain enzymes or certain proteins that they have been known to be responsible for resistance. Then it's also going to recognize predicted resistance profile, predicted resistance profile, and it's also going to tell you the lineage. For example, from lineage L1 to L4. So L2 is from Beijing, which is from China. If at the end of the day, the TB profiler now now gives you a data that is on L2, that is to tell you that your TB is a native, is from China, is from Beijing. So for that, okay. Now, optionally, that is, after you must have used TB profiler, you can cross-check with microbe predictor for consistency. You know, I told you that you can run the you can make use of chemomatic, fastp, or you make use of COTS adapt. So you can make use of it in order just to have a kind of consistency. I said you can use your TB standalone tool, or you can use the one from Galaxy. So it is better for you to use both of them so that you'll be able to compare the consistency. Okay. Now, what are the rationale? What are the reasons for you making use of a TB profiler? It is a specialized pipeline for TB whole genome sequence, which is widely used in resistance and lineage prediction, and it is recommended. So if you are dealing with Mycobacterium tuberculosis and you do not TB profile it, honestly speaking, your work might not be that too acceptable, because it is an internationally recognized tool that they use to profile. That's why they call it profile. If somebody say that they need your profile, they need everything about you, including where you are from, including what you can do, including every information and characteristics about you. So that is why TB profiler is very, very useful for you. Okay. Now, the next thing is we have actually talked about this. That is number 8, which is genome assembly and annotation. One thing is to assemble. That is, you have information about Mycobacterium tuberculosis. Another thing is to be able to annotate them. Annotation means that you map. Okay, for example, if you say, oh, you have one finger. If you are annotating it, that means what are you using that finger to do? That is annotation. So if you see any gene, and the research has told us that up to today, more than 60%, of proteins in Mycobacterium tuberculosis are still, their functions are still not known up to today. So by the time you will now get to that place, there are some gene, or there are certain proteins that you will not be able to annotate them, even if you will not be able to know what function they perform. So that is that. So, and then another thing is one of the reasons why you must annotate, you must assemble as well as annotate your sequence, is that number one, it will help you to detect structural variations. So don't forget, that the tail in human beings, as I said, it has disappeared completely. In evolution, it was not one day job. It was not one day job. It is a journey of more than thousands of years. Don't forget that um, before before you will see an organism that is developed now, before you see it and you see the everything, you see the structure. That structure was not there just by one day. It's not just one day. Let me tell you one thing. Let me tell you one thing. Before all of us decided that we needed to be putting on clothes, Oṣejọ kan, not one day. There was a time, and even you now, you know, even if anybody do not tell you, there was a time that your parents will be looking, so why was Shoni? So the shaking conning, just leave me alone, because you thought you have seen it all. That time, your this thing will not come that, oh, you should not be walking naked. So what we are saying, yeah, is still the same thing, that any organism you see today and any structure that you see in any organism today is not just one day. Not just that, it's not just appearing one day. There was a time that they did not even know the function that structure was meant for. Huh? So until they started annotating it, they now link it with this function, they link it with another function, and they now say, yes, that is what is called annotation. Okay? Then you should be able to perform pangenome style presence or absence of analysis of immunogenic genes. I told you, when you, for some of us, that will be working with vaccine design, and I've told you also in immunology that if so, there are some substances, there are some chemicals that they are antigenic, they are antigen, but they are not immunogenic. Antigenic means that they will be able to trigger an immune response. Antigenic, that is antigen. What are antigen? Antigen are foreign substance. So that means before you will be able to design any vaccine, the protein you are interested in must have the capability to trigger an immune response. Because if no immune response is being triggered, what do we call vaccine or vaccination? Vaccination is just teaching you, training your body that in case you have, in case you come across this microorganism, let me prepare you ahead. It's just like your parents that is telling you that, my dear child, my dear daughter, see, it is not easy to cope with men, but anytime you come across any man, this is how you should behave. That is just like the same thing that your vaccine is trying to do. Your vaccine is training. For example, if you are developing vaccine against Mycobacterium tuberculosis, you are not going to take substances from another organism. You are taking vaccine from that Mycobacterium tuberculosis that is causing that problem. That is where you are getting that protein from. So that by the time you use it, you now inject it, it is going to trigger and prepare your body ahead that, oh, let me prepare your immune system in case you encounter Mycobacterium tuberculosis or any similar gene to this. So that is what this annotation will also tell us. It's going to tell us what are those genes that are immunogenic, that have the ability to trigger an immune response. And sometimes you can have a protein that is very, very essential, but it might not be antigenic and it might not be immunogenic. Just discard it. You cannot use it. That's the reason why when you retrieve like 35 sequences, for example, there will be a point that you will pass it through, you will filter it. Once it cannot meet up with that condition, drop it. It doesn't mean that that protein is not useful. It is also an essential protein, but they cannot be used for vaccine development. So that's the reason why that as I told you that you needed to know everything, almost everything, the behavior. For example, now if you want to conquer, you want to conquer your enemy, you must know almost everything about your enemy. When your enemy is going in, when your enemy is going out, you must master almost. You, in fact, there will be some times that you also have to behave like them. You have to mimic them so as to make sure that. So that is just the same way, and that is what genome assembly is going to do for yours and annotation. Okay. So and the tools we are going to make use of is spade. We have used spade, abi? Spade for de novo assembly. If we are not using de novo assembly, that means we might not use spade for it. Okay, then we also use proca for annotation. In fact, the one we are going to use now is going to be that proca. Please let us go to um let's go to Galaxy. Go to Galaxy and type proca. Is there any? Can I go on? Hmm? Timileyin, what is it? Hmm? What disconnects? Are you sure? Has it has it disconnected? Because my own is still on. Proca. So just type proca. So proca is used for prokaryotic genome annotation. So just like I told you, one funny thing about a galaxy, since this galaxy is all these tools are inside Galaxy, is that the last procedure or the last um, task that you perform automatically is going to be available for the next one you are going to make use of. Okay, so when you get to Galaxy, just type proca and go and click on a prokaryotic genome annotation. So when you get there, click on multiple data sets and also click on browse and this. So the last one here is what is the problem? Hmm? It's not running. Okay. So have you typed procar? Okay. So already just click on that what do we call this? Okay. So please just go there and look at that down this. So 38, 37. Just check where you have spade. Where you have spade, that is all the one you are going to run. Okay, selected. Spade. So, and leave every other thing, leave it. Spade. Spade is check here. check by the side. Okay, you have not done the thing. Okay. So run, I thought you have run straight. Okay, please try watch this one. When I'm going to give you time so that you will go through it. So now, immediately the spade will make sure that you have contigs, and you have scaffold. Contigs and a scaffold. What contigs means, I'm going to let us consult with ChatGPT ChatGPT on what contigs mean. ChatGPT